Really quickly, for those of you who don't know about the Build Exito program, this is a really wonderful, actually quite a lot of money too, way to train researchers, particularly those from disadvantaged groups, if you're at all interested in looking into this program. It says, I guess here, 9 days until the applications are due. It's actually only 5 because I forgot to spread this email to you before then. I will put this link here for you to check out on the website, the D2L website. I also have the information on my Mutant Viruses from Hell lab up there. A couple of people have talked to me about that already. I think Monday is the day you can start registering for classes. So I wanted to put that out there. Yes, I'll be teaching virology next term. And one of the things that I only literally just noticed when I was looking at this, this is actually Diana Demchenko, who's working in my lab, and this is one of the viruses that we work with. So yes, I also work with this Exito program. One other quick thing, I know you're not thinking that far ahead at this point. On the 23rd of February, i.e. the day after our second midterm, OMSI Meet a Scientist. I don't know how many of you know about this program. It's a lot of fun, mostly for slightly younger people than you. But I'll be there having my Build a Virus demo. One of my postdocs will be there with a virus evolution demo. One of my other colleagues from the biology department, Dr. Annie Lindgren, will be there talking about deep sea macroorganisms. So that should also be a lot of fun and a nice way to decompress after the midterm, I think. So hopefully that will be the case. So with that being said, let's, oh, sorry, yeah.
I didn't see any posting for the last lecture.
You did not see any postings for the last lecture. That is correct. There was not a posting on YouTube for the last lecture. I know somebody at least took a audio recording. I've got the last 2 minutes. I could post the last 2 minutes. But I have the upgraded software now. So hopefully that will work and we'll get something. Part of the reason I haven't linked to last year's lecture is because I had a guest lecture last year for this subject matter. So that's why that's not there. But if somebody has an audio recording, please send it to me and I will post it. So that will be there. Apparently with the new software, I can also input PowerPoint. So maybe I can try and put some of the slides together with what I'm saying. We shall see. I'm going to make no guarantees whatsoever about that. But hopefully, I've also got a backup recording today, my own audio. So at the very least, we should have an audio recording of today's lecture. But today, wanted to talk about what happens after transcription. So that's why I don't have my model here anymore. I don't have a nice RNA model. Don't worry, the DNA model will be back when we talk about regulation later on. But this has to do with what happens to messenger RNA before it gets translated. And the easy way to think about this is actually if you look at prokaryotic cells, bacteria and archaea. These are way more streamlined. And in fact, the whole point of a prokaryote is it has no nucleus. And so what that means is that transcription and translation are taking place in the same compartment. Yes, it's biology. There are always exceptions. But for the most part, they're all happening in the same compartment. What that means is as the RNA polymerase is transcribing and making the messenger RNA, translation actually happens at the same time. It's what's called a co-transcriptional translation. So they're happening simultaneously. That's not what happens in eukaryotic cells, which have nuclei. So in eukaryotic cells, you have transcription that takes place. And we've talked a lot about this already. The vast majority of the DNA that gets transcribed and made into RNA are these introns, which actually are not protein coding, that you don't want to have in the final version of your messenger RNA. It gets translated out here in the cytoplasm. So there are multiple different steps. We'll talk about 4 of them today and on Monday. First step that happens, going from 5' to 3', again, not surprisingly, because that's how they're being transcribed as well. You have 5' capping, RNA splicing, 3' polyadenylation, and then RNA export out of the nucleus and into the cytoplasm, where translation takes place. Again, always exceptions to some of these things. There's maybe a little bit of translation that happens in the nucleus that's a little bit controversial whether that's happening or not. But let's talk about that process now. And again, not unlike what happens conceptually in the prokaryotic cells where you have transcription and translation happening simultaneously. Actually, transcription and processing happens simultaneously as well, at least these first steps, capping, splicing, and tailing, as I'll usually call it, for polyadenylation. That's happening as the RNA polymerase is making the RNA, as that RNA is being released from RNA polymerase II. And partly how that happens is that we have all of the regulatory factors, I should say the processing factors for messenger RNA that are associated with RNA polymerase II. And I'm sure as all of us remember, we have a extension on the largest subunit of RNA polymerase II, also known as the C-terminal domain, which gets phosphorylated. What does it get phosphorylated by? Kinase is what particular kind of kinase? TF2H. No, but you haven't reviewed that part for the midterm yet. No, one of the general transcription factors, RNA polymerase general transcription factor, TF2H, has helicase activity. That's what the H, well, actually it's not what the H is for, but it's an easy way to remember it, and kinase activity. So it's phosphorylating the C-terminal domain of RNA polymerase II. And that's important for both getting away from the promoter and also for binding for a lot of these proteins that are involved in capping, splicing, and 3' end processing or tailing. So this process, we're going to talk about them individually. We'll talk about capping separately from splicing, separately from tailing, but they're all happening pretty simultaneously as the RNA is being made. And it turns out that's actually particularly important from a splicing point of view. So what do these mRNAs look like? Bacteria, again, pretty straightforward. I have bacteria here rather than prokaryotic because there's actually some differences in archaea that we're not going to talk about. But because transcription and translation are happening at the same time in the same place, you have a triphosphate at the 5' end of your messenger RNA because that's that first nucleotide that the RNA polymerase put down. That is extended 5' to 3'. In many cases, but not in all, this RNA can code for completely different proteins. So here just shown as alpha, beta, and gamma. This is what's also called polycistronic. A cistron is a protein coding part of your messenger RNA. So polycistronic, very common in bacteria. This is also a very efficient way if you want to regulate a whole bunch of protein coding genes at the same time in the same way. And we'll talk a lot more about that later on. Eukaryotic messenger RNAs, however, are a mess, at least as far as I'm concerned, being very microcentric here. So eukaryotic messenger RNAs also have a triphosphate at the 5' end, but it's not free and floating around. It's got an extra guanine and a methyl group that's attached to it. And this is the 5' cap structure. We're going to talk a lot about that 5' cap structure later on here, how that actually gets made. The main purpose of that cap structure, actually there really seem to be kind of two main reasons for it. One is it protects the ends from getting degraded by all of the hungry exonucleases. Lots and lots of extra exonucleases floating around in the nucleus, floating around in the cytoplasm. So having that cap structure is really nice for protecting the end. The other thing is it provides a binding site for proteins that are going to be involved in both getting that messenger RNA outside of the nucleus, but also involved in translation, when we talk about translation later on. So that cap is a really important structure to have export and translation purposes. Here, we don't have all the splicing going on, but the vast majority of eukaryotic messenger RNAs are spliced in some way, particularly in the larger eukaryotes like us. So this is often split up into different pieces. We'll also talk about some of the alternative splicing. These messenger RNAs can encode a bunch of different proteins, but they're all slightly different proteins. That's because that piece of RNA is being mixed and matched in different ways. This is usually thought of as a monocistronic RNA. Another thing here in the light blue, hopefully obvious here, these are what are called non-coding sequences, or UTRs, untranslated regions. And we'll talk a lot more about UTRs when we talk about translation later on. Turns out they're also very important for regulatory mechanisms. And then, should not be new to any of you, poly-A tail, between 150 and 200 in most cases, stretch of A residues that's attached after you have regular transcription and before you have termination. A lot of this poly-A tail actually has to do with similar kinds of functions as what the cap is. It binds to proteins and also protects the end of the messenger RNA from getting degraded by the exonucleases. So let's start looking at the 5' end here. What do we have? Cap. Well, the cap is put on after you have transcription. And RNA polymerase II, let me get this out of the way here, sorry. Don't need this quite yet. 5' end of your RNA, as it's being made by the RNA polymerase, is a triphosphate, because that's the first nucleotide that gets put down. Again, RNA polymerases don't need primers, so big difference between DNA polymerases and RNA polymerases. The first thing that happens is that last phosphate, this would be the gamma phosphate on this triphosphate, gets cut off. A phosphatase literally just chops off that last nucleotide because that needs to be the substrate for one of my favorite enzymes, the guanylyltransferase. Say that 15 times quickly. The guanylyltransferase, which takes GTP and hydrolyzes the pyrophosphate off of the GTP and hooks up that last phosphate, the alpha phosphate, to what would otherwise be the beta phosphate here from that original nucleotide. What this does is it gives you a 5' bond. So literally what's happened here is you've taken what you normally have, here again 5' to 3', that's the way everything is hooked up when you have normal polymerization, but now you flipped it around. And again, that's probably having to do with protecting it from the exonucleases, which would otherwise love to chew in at the ends. So that gets flipped around. The guanylyltransferase makes a 5' phosphotriester because there are 3 of these bonds here, also the triphosphate bridge. After that happens, you have to add a methyl group to this guanine. Comes in as a regular guanine, but then there's a methyltransferase that puts a methyl group on here. This is your cap structure. This cap structure gets put on after about 25 nucleotides have been transcribed. And it turns out that 25 nucleotides is just about enough nucleotides to get outside the RNA polymerase complex. So as soon as that end of the newly made, or also here it's called the nascent RNA, comes out of the RNA polymerase, the cap modification enzymes jump onto it and start modifying it. In some cases, but not in all, you then also have methylation of riboses on cap structures. And part of the reason that I mention this is that sometimes people will talk about cap 0, cap 1, cap 2. Cap 0, cap 1, and cap 2 just refer to the number of methyl groups on riboses. So if you have no methyl groups on your riboses, so not here and not here, that will be a cap 0. If you have 1 methyl group, that will be a cap 1. If you have 2 methyl groups, that's a cap 2. Has to do with actually slightly different caps, how they're being translated, etc. But for our point of view, cap 0 is all we really care about. And again, it's got a methylated G with a 5' to 5' triphosphate bridge at the end of your primary transcript. That again gets bound by proteins like the cap binding complex and again in translation. So that's it for the 5' end. What do we have next? No, a clicker question. Okay, so go forward, forward, click, forward, back. There we go. How many different enzymatic activities are required minimally for the formation of a cap on eukaryotic messenger RNA after it has been transcribed? 1, 2, 3, 5, or greater than 5? Stedman loves these kinds of questions because it's really easy to come up with the wrong answer. Ten, 5. Still going for 100%? No, we're not quite at 100%, but we're close enough. Above 80%. So you can talk about this amongst yourselves if you really like, but obviously it's pretty clear. What are those 3 enzymes? We've got a phosphatase, we have a guanylyl transferase, and a methyltransferase. You could also have the ribose methyltransferases on some caps, but this is the minimal case for caps. So now we'll talk about splicing. I thought I'd throw in some of these extra slides, so just in case you're thinking about studying for a midterm, it's easy to split up what things are where. So before we talk about splicing, however, I wanted to talk about the way that splicing was actually detected. And this is a completely artificial technique, but an incredibly useful one called the R-loop technique. So R just stands for RNA. And the whole idea of an R-loop is you have a processed, proper eukaryotic messenger RNA that you then hybridize back to the double-stranded DNA that it started with. So hybridization, again, we've talked about hybridization before. You heat things up, treat them with various chemicals, put in your RNA, let those strands come together, and what will happen is you will end up with a double-stranded piece, which is where your RNA is bound to the template DNA, and then a displaced DNA strand. So how do you see this? You use electron microscopy for this. Maybe a little bit hard to see here, but basically what you're looking for are where you have thick lines, like you have down here at the bottom. That means it's double-stranded. And thin lines where you have something which is single-stranded. A lot easier here when somebody draws out the cartoon. This is your RNA, which is double-stranded here. It's got this little piece hanging off that actually is not going to be hybridized into anything. Any guess what this might be? Poly-A tail, which is not something which you can hybridize to. And in this case, and this is partly how this process was discovered, there's a piece of RNA which is hybridizing to yet a different piece of DNA that's not contiguous with this DNA right here. So another way of looking at this, and actually a case where you don't clearly have splicing, here's a 3' end of your RNA, hybridizes to your DNA, displaces this single-stranded piece of DNA, and you have a short 5' piece here. So what people did, and when I say people here, it's basically Rich Roberts and Phil Sharp and their groups in the late 1970s, was they purified lots of RNA and then hybridized it to various different DNAs, and they saw some really weird things going on. And particularly that was true with, oh, hang on, we've gone one too far here. No, there we go. With a really wonderful source of lots and lots of DNA and RNA that's really perfectly identical, many, many copies of it, relatively short, not too complicated. What are you going to use? What's the course that Stedman's teaching next term? Viruses. So splicing was originally found in viruses, and actually, originally, these researchers thought it just happened in viruses. And soon thereafter, they actually showed that it was happening in normal, i.e. non-viral eukaryotes as well. And so this is the colored image. It was, of course, a black and white micrograph, which showed one of these R-loop structures. And in fact, this was an incredible R-loop where we've got the double-stranded, this is the RNA here in black, which is double-stranded, and the rest of it being green here. And what has happened in this R-loop process is in these big loops of DNA that don't match the RNA. So big loops of DNA that don't match the RNA. How did that happen? Well, pieces of the RNA actually got chopped out. And so it was in this mechanism, say this process, where they saw in the case of adenovirus, and we'll talk a lot more about adenovirus next term, there are these three sequences, also known as the leader sequences, 1, 2, and 3. Turns out that these are attached to almost all of the transcripts, which are the final transcripts that the virus uses to make more of the virus. So these sequences, 1, 2, and 3, are always together, and the only way they can be together in the RNA is if you loop out the DNA in between. So loop out of A, loop out of B, and then there's this big loop of C, which then is to where you have the protein coding sequence, and this protein coding sequence is the main part of the capsid, also known as the hexon protein, that again we'll talk about much more when we talk about these viruses next term. So a lot of this came about because people now knew what sizes all of these pieces of DNA were. They had to actually figure out these sizes by agarose gel electrophoresis, which was developed right around this time. But even more importantly, they needed to chop up their DNA into different size pieces. How did they do that? Restriction endonucleases. So restriction endonucleases, Rich Roberts here, who was at Cold Spring Harbor at the time. Cold Spring Harbor has just stripped Jim Watson of all of his titles there. Good job, well done. Rich Roberts was making all of these restriction endonucleases and basically giving them to all of his buddies, and then said, actually, there's a lot of people who want these. I'm going to start a company. So he actually works for New England Biolabs, which is one of the major suppliers of restriction endonucleases for researchers really throughout the world. So Roberts and Sharp basically independently showed that this was what was happening. Both of them in adenoviruses. Again, they both got the Nobel Prize here separately for doing that. Turns out that it's not just in viruses. It's in all kinds of eukaryotic cells. And long story short, introns are incredibly variable. We talked about this before. Introns can be a couple of nucleotides long, actually minimum's about 40 or 50, but they can be hundreds of thousands of nucleotides long. Exons, on the other hand, are almost always about 150 nucleotides in length. And that is hopefully what you can see up here. Two different human genes, the human beta globin gene, the human factor VIII gene. Any of the vertical red lines, they're kind of hard to see here because there's not much in the way of introns in the beta globin gene, whereas there are lots of introns here in the human factor VIII gene. Each of these vertical red lines are those exons. And those are what gets put together. You lose the introns in this splicing process. So how does splicing work? Turns out you need 5 RNAs and a whole bunch of proteins. I put 100 proteins here. In rereading the textbook, they say more than 200 proteins. And we'll look at some of the images of some of the higher resolution structures. There are a heck of a lot of proteins that are involved in here. But one of the interesting things about splicing, when you do splicing, what are you doing? You're cutting DNA, or RNA and religating RNA. And that process in and of itself is isoenergetic because you're cutting a bond and making a bond. So there's no actual energy input which is required for that step. Turns out, however, that splicing is incredibly dependent on ATP hydrolysis. It actually requires lots and lots of energy. And it turns out that the energy that's required is really for specificity. It's for making sure that you have the right splice at the right time. Because if you look at just a nice example here, again, of the human factor VIII gene, there's only a few bits that you want, these red bits. All the yellow bits you don't want. So you want to be very sure that you're cutting out the right pieces and keeping the right pieces. So how does this work? It's another weird kind of bond that happens in RNA. If you remember, RNA is different from DNA in two different ways. We've got uracils, but we also have an oxygen at the 2' position of the ribose ring. And that's critical because what happens is as you have splicing, the first thing that happens is this 5' exon cuts here. Now you have an OH at the end here, but the 5' phosphate, this now attaches to a 2' OH in the intron, what's also called the branch point. This, and it's always an adenine residue. Then this lariat structure with the 5' 2'. So we had the U4, U5, U6, and all of these RNA-RNA interactions, they're base pair interactions. So how do you break apart base pair interactions? Helicase activities. Is this DNA or RNA? It's an RNA helicases. So the RNA helicases, this is where you burn all of your ATP. It's in the activity of the RNA helicases, pulling those RNA strands apart, and then having them come back together, pulling them apart, putting them back together. And we'll see this in much more detail in the next couple of slides here. The first one, which is the ridiculously oversimplified version that we have in the textbook, is here, where we have interactions between U1, which is at the 5' splice site. This is the 5' end of your intron, forms these base pair interactions. Then the activity of RNA helicase using ATP hydrolysis, these base pairs get exchanged for these base pairs on U6. The same thing happens when you have your branch binding protein. Now this is not actually a RNA helicase activity directly. Here it's just removing this. As I mentioned, this is that's the total oversimplification that we have in our textbook. In the last few years, there's been a lot of progress in terms of understanding the three-dimensional structures of a lot of these RNA molecules, and particularly the RNA together with protein in the spliceosome and all kinds of other zomes, the ribosome. When we talk about the ribosome, we'll look at this in much more detail as well. So this is a paper from, I believe, 2015, where they looked at the structures of the different RNAs and where they are relative to the branch points, etc. So cartoon version up here at the top. Again, we have the U2 RNA. Most importantly, the purple here, that's where our intron is, and the U6, which has that enzymatic activity. And you can see where U6 is right next to where you have the G, again, which is the 5' end of your intron, and this A, where you have your branch point right here. So lots of base pairing with U2 and your intron, lots of base pairing with U6, with U2, and also with the intron structure. The big thing that they found in this particular structure are these metals in red, which are what seems to be catalyzing the bond formation, again here, between the 5' and the 2'. It's really nice to draw these strands just as nice lines, but of course they're not. They're helical. They're wrapped around each other. And so these are what they actually look like. Again, same color scheme. Purple is your intron. Green is U6. Blue is U2. And the take-home message that I get from this is, wow, there's a whole bunch of crazy base pairs interacting here, and clearly to do anything else, you've got to pull those base pairs apart and put them back together. So this is the zoomed in version of that. If you zoom out a little bit, also in the same paper, this is hopefully giving you a bit of a view where those RNAs are. In fact, it's actually kind of hard to see where the RNAs are here in this structure. And this emphasizes that there are lots of proteins also involved in the spliceosome. Here are U5 is in blue, U6 is also in blue here, U4 and U6 base pairing here. So all of the RNAs are in blue. All the proteins that are associated with this are in various different colors. And they kindly put this together in a version where you can see them a little bit more easily. This is U6 and U4 associated with each other. This, I should, back up a little bit here. This is actually before you have splicing. So this is when U4, U6, and U5 come into and bind to your intron that has U1 and U2 already associated with it. And a lot of that bringing in of, mostly it's U6 because that's where we have our activity. That's being brought into the complex, but in order for U6 not to be chopping up or splicing together any old random RNAs, it's got to be kept in an inactive form. How is it kept in an inactive form? It turns out it's kept in an inactive form because it's base paired with U4. So the U4 base pairing with U6 allows that complex to associate with the rest of your splicing complex. Then you have RNA helicase activity that pulls them apart, rearranges the process, so then you have U6 in a place where it can actually go through the splicing mechanism. So here, even more detail. Now we actually have our intron together with U6, see, very closely associated with each other here, and all of the other RNAs in the context of all of these other crazy proteins. And this was, just from 4 years ago. We've gotten more and more structures as time has gone by. But the keys here is the RNAs that have the activity, RNAs are giving your specificity, and the RNA helicase activities, which is in fact what a lot of these proteins are on the outside here. All of these different proteins are involved in either putting the RNA in the right place or has the ATPase activity to pull those strands apart. So no, I'm not going to ask you all the names about these things. The only things we need to care about are the ones that were back on that earlier slide: branch binding protein, U2AF, and then the various RNAs. Questions about this, or we're overwhelmed with the structures? Overwhelmed with the structures, yes. So that's why I wanted to emphasize the activities of 1, 2, 4, 5, and 6, and then branch binding protein U2AF. Questions, comments, worries? Ready to answer a clicker question? Yes? We'll get 80% the first time around on this one too? Good. Which of the following is thought to contain the splicing enzymatic activity? Another ribosome or the spliceosome? Come on. I should write these things properly. Spliceosome. Spliceosome, not ribosome. Ten, 5, Okay, we didn't break that 80% barrier, so tell each other what you selected and why. Give you a minute. That's what he said. He's got to be right. Ready to get 100% on this one? Yes, let's go. Again, feel free to continue to discuss here. Ten, 5, 2, 1. Stop it before I show you the results. So yes, it is U6, the small nuclear RNA. U4 seems to be regulating the activity of U6. U1 is bringing us to the 5' end. U5 is helping out to add that complex. U2AF is just showing you where to put U2 onto the rest of the spliceosomal complex together with your intron. So the immediate question I'm sure all of you are asking yourselves is, why aren't there more mistakes? Because if you remember those consensus sequences, they are not very good consensus sequences. You find GUs and As and AUs all over the place in your genome. So how does the cell know what to use in terms of what's an intron and what's an exon? So you'll remember that the exons have pretty conserved lengths, again, about 150, 200 nucleotides, whereas introns can have incredibly variable lengths. So that's one kind of giveaway here. The second one actually is, as we talked about right at the beginning, we'll talk a little bit more about in just a second, because you've got all of those splicing proteins and RNAs, not surprisingly, attached to the C-terminal domain of the RNA polymerase. Splicing happens as the RNA is being made. So you're going to start making the splice site as it's coming out. So one of the things that's relatively rare is what happens here, the whole idea of exon skipping. So if you go from exon 1, your 5' end of the intron here, instead of going to the 3' end of the intron here, it skips and goes over to here. But the fact that you're doing this in a co-transcriptional process means that you're most likely to get this and this one put together, because you haven't even gotten to this exon yet, because it hasn't been made yet. So that's one of the things that happens. So just the co-transcriptional process. The other thing, and probably even more importantly, are the presence of these so-called SR proteins. Now, why are they called SR proteins? It's got a bunch of serines and arginines that are associated with them. And arginine is one of those ones I asked you to remember. What's the important thing about arginines? Positively charged. What's the charge of RNA? negatively charged. Although these things seem to bind not just through charge-charge interactions, and there are a whole bunch of different ones. And that's supposed to be shown here by the fact that we've got circles and crescents and ovals, etc. So lots of different SR proteins. But SR proteins bind to exons. And part of that binding to the exons, the SR proteins really often interact with not so much U2, but U2AF. And U2AF is that first thing that binds to the branch point. And one thing you may go, well, wait a minute, isn't the branch point somewhere in the middle of your intron? It turns out that branch points are almost always very close to the 3' end of the intron. And that's because you've got these SR proteins that are interacting with U2AF, which is bringing in U2, which tells you where the branch point is. So very rare that you have the nice lariat structure with a long tail on it. It's usually mostly loop with a really short tail at the end of it. So that's probably the major reasons, the presence of these SR proteins. There also are a number of proteins that bind into introns, and these are very confusingly named heterogeneous nuclear ribonucleoprotein complexes. Heterogeneous means they're just a funky mess of different proteins. But it turns out that they're really important for RNA export a little bit later on when we get there. But it's really about where you have U2 and this branch point and where you're going to be assembling the whole spliceosomal apparatus in terms of where you end up getting this splice. And so one of the things that happens is there are also what are called these cryptic splice sites, and all that cryptic means is one that's not used all the time, and that could mean that you end up splicing in the middle of an exon as opposed to at the end of an exon. Very rare that you're going to have a cryptic start site here differently. Occasionally you will get that as well. Another thing which helps out a great deal with these is that exons are almost always the same length. This is kind of a broken record here. 150 to 200 nucleotides. As all of you remember, I'm sure because you're studying for the midterm for next week, that nucleosomes also packaged about 150 nucleotides. So probably also nucleosome association, sort of one nucleosome to one exon, is a pretty good rule of thumb. And that's probably why you have exons being confined to that more or less the same size. And so this is just a distribution of the different lengths of exons found in the human genome, the C. elegans genome, and the Drosophila genome, all of them around 150. On the other hand, if you look at introns, they're all over the place. Some are really short. In fact, in humans, you have very few short introns, but in both C. elegans and fly, quite a few short introns. These genomes are a lot smaller in terms of numbers of nucleotides in them, and you have longer and longer introns, particularly in humans, again, some of them over 30,000 nucleotides. So why bother with having this ridiculously complicated structure to have an RNA and get rid of, in most cases, 90+% of it? Why does this make sense? And why? The answer to any kind of why question in biology is evolution. And because is also not a bad one either, but evolution is what I was looking for. And so why seems to be that, well, because you're cutting out all these pieces, you can actually cut out some pieces in some cells and cut out other pieces in other cells and end up with variants on your proteins just based on which exons you use. And this is what's called alternative splicing. I used to have 60% in here. Previous years, the textbook says actually more like 95% of genes in humans are alternatively spliced. And so that means you can have all kinds of slightly different proteins. And very often these would be, say, different domains in a protein. One protein will have a particular domain, another protein will have a different domain. We talk about regulation of this later on. We'll talk about exactly what happens in some antibody cells and where these end up going. But for this purpose, basically the presence or absence of different exons will give you a slightly different protein, which will give you a slightly different structure, which of course will give you different activity in or function in these different kinds of cells. So smooth muscle, striated muscle, fibroblast, brain, etc., all have this same protein, but has slightly different exons, and it's going to have slightly different structures. The problem with alternative splicing gets back to that whole wonderful consensus sequence that we had, right? Consensus sequence, which is kind of crap because you find GUs and AUs all over the place in the genome. If sometimes they're used and sometimes they aren't used, just by looking at a DNA sequence, it's going to be really tough to figure out what that final protein and that final RNA is going to be. And this is why people love cDNA libraries. Because the cDNA library, you've taken the final processed messenger RNA, and then you made a DNA copy of that and figured out what it is. So you use the cDNA library to figure out what the splicing is and how that splicing took place in any particular cell, because you've isolated the RNA from fibroblasts, for instance. And in fibroblasts, you find that the cDNA has all of these particular exons in it. If you isolate messenger RNA and make cDNA from muscle, it has different ones that are in it. And as far as I know, the extremist case, extremist, is that really good? Most extreme case of alternative splicing is actually in Drosophila, protein called DSCAM1. It's not that important, but the alternative splicing that you can have in DSCAM1 is a multiple of these 12 alternative exons by 48 alternative exons by 33 alternative exons by 2 alternative exons. You multiply all those together, 38,000 different possibilities for one gene in terms of the RNA that it makes. Turns out that this is really important for the immune system of Drosophila, so having lots of variability is really, really important. Another thing I wanted to point out here is that in most cases, and you can see it here as well for this tropomyosin gene, most of the exons are used. All of these gray exons are used in every single one of these 38,016 possible ones. It's just you have a few alternatives. In this case, you have 4 different groups of different alternatives. But also, again, if you look over here, most of the time, these are going to be, this exon is conserved, this exon's conserved, this exon's conserved, this one's conserved, this one's conserved. Just a few of those different exons are used or not used, as the case may be. So that's alternative splicing, and we'll talk about how you get alternative splicing a little bit later on. These are just a couple of extra images from more recently looking at the structure of the spliceosome. Again, U6 interacting with your intron, having its catalytic activity. And this, I think, is the latest version that I saw from 2 years ago, again, looking at where all of these RNAs are inside the rest of the proteins. So sort of a review of splicing to now. Co-transcriptional. Turns out that also how fast you're transcribing is going to make a difference in terms of how much alternative splicing you have. So as I mentioned, when we looked at that first alternative splicing thing, skipping exons is much less likely to happen if you're transcribing slowly than if you're transcribing quickly. Because if you're transcribing really quickly, you'll have a chance to have that extra exon that you could end up splicing to. Another thing that seems to make a difference is histone modifications. And so the DNA which is being transcribed has modified histones that are attached to it. Some of those modified histones are interacting with proteins that are important for getting alternative splicing. So let's look at that process here, how you get some exons and how you don't get some exons, and I should say how they're used or not used. We'll talk a lot about enhancers and silencers when we talk about transcription later on. Now we're talking about splicing enhancers and splicing silencers. So anytime you talk about an enhancer or a silencer, this is a sequence that proteins are going to bind to and give a particular activity. This is just a slightly different way of looking at exons. So here's an exon. In these big boxes, here's the intron, the thin line, exon, intron, etc. If you have an splicing enhancer, what splicing enhancers do is they help the binding of these SR proteins. Here they're called RS, just to confuse you. Again, it's arginine and serine-rich proteins. These guys, the enhancers, help these SR proteins bind to exons. If the SR protein binds to an exon, it binds to U2AF, which binds to U2. You assemble the spliceosome. If, however, you have a silencer and a protein that binds to the silencer, which happens to have a lot of glycine residues rather than serine residues in this particular protein, that will block the binding by the RS protein. You're not going to have SR proteins. Doesn't help binding of U2AF. You don't get U2 binding, so you skip that exon and move on to the following exon. Just in case this wasn't confusing enough, there are multiple different ways that you can have alternative splicing. Again, this is partly why it's really hard to predict what kind of splicing you're going to get just by looking at DNA sequences. In some cases, you'll just skip exons. That's what you have here. You can have a different 5' splice site. So this is spliced in the middle of an exon. Different 3' splice sites. Some cases, you actually hang on to introns completely. Sometimes if you splice to one exon, you won't get the other exon, and vice versa. Sometimes, depending on where your promoter is, you'll end up with some exons that will be made, some exons that won't be made. And in some cases, you actually end up with alternative poly A tails. So splicing and this alternative splicing can give you all kinds of different outcomes. And all going to be dependent on where you have the assembly of all of these proteins, how they're all interacting with the exons, introns, etc. And not surprisingly, this actually turns out to be a big problem with genetic diseases. So one of my students found out probably about 15, 20 years ago, as I was working in my lab, had incredibly doughy skin and wasn't clear what it was. Eventually, he was diagnosed with Ehlers-Danlos syndrome, which is a problem with collagen. Collagen, turns out, is one of these genes that has a ridiculous number of exons in it and all kinds of splicing going on. He actually had a mutation in one of those splice sites, which caused his collagen actually not to be made, and he had a ridiculously flexible skin. So it turns out that a pretty high proportion of genetic diseases, particularly those that happen because of single nucleotide changes, and we talked about with DNA repair, anytime your genome is replicated, there's going to be 5 to 10 nucleotide changes that have happened. Those happen to be in places where you could get different splicing. Quite often you'll have some kind of genetic disease. And the case here is beta thalassemia. And in fact, I had a master's student who had a similar kind of mutation to this. And what's happened here is there was a mutation at this position, actually in an intron, which ends up giving a new splice site here. And so you have a shorter intron, and that means you have extra coding sequences which end up here. Sometimes you'll have a single nucleotide change. This is the kind that she actually had. You can also have a single nucleotide change that will get rid of one of the either the branch binding site or the 5' end of your exon. And you can even create completely new ones. So these splice sites can cause disease, but also could be a way that you're getting evolution. So why? Evolution. So mixing and matching, adding extra pieces of DNA to a particular protein. If that works, it's maintained in the offspring. If it doesn't, it gets thrown out. So another way that you can have this. So the whole process of doing all this splicing is, again, ridiculously inefficient, but at the same time allows a lot of changes and evolution, but unfortunately can end up causing a number of diseases. So just wanted to finish up today anyway with, as if it wasn't complicated enough already. Most splicing happens here with the major spliceosome, U1, U2, but there are alternative spliceosomes as well, which have different small nuclear RNAs that bind to different sequences. The process here is almost identical. It's just the recognition of these different sites is different. And it turns out that in a very few cases, you can actually take different RNAs and splice them together. And so it's what's called trans-splicing. Again, the mechanism here is very similar. You just have to have binding of particular proteins to particular places. And we're not going to have the last clicker question today. We'll have that starting on Monday. So something to be prepared for on Monday morning. Have a good weekend.